Okay, we're now in the third part of the overview from the textbook dealing with protein synthesis, and part 3 is all about the ribosome and what's happening in the ribosome. Figure 31.14 shows and the structures of the different portions of the ribosome, the 50S subunit and the 30S subunit. These come together to form the complete ribosome with its decoding apparatus here in the 30S subunit, and the chemistry of peptide bond formation up here in the 50S subunit. Figure 31.15 shows an illustration of all the secondary structure elements, all the base pairings, the stem loops in the 16S ribosomal RNA. This is a portion of the 30S subunit, and the 16S ribosomal RNA is responsible for decoding codons in the messenger RNA, making sure that codons and anticodons match up exactly with each other. And here's this emphasizes the base pairings, but it's actually a three-dimensional entity, and this is this panel B emphasizes that three-dimensional form. Okay, so there are 3 special sites on the ribosome, so more complicated than a regular enzyme. There's 3 positions to accept these very large substrates. There's an A site, which is the place where you find the amino acyl tRNA. This is where you have an activated amino acid coming into the picture for protein synthesis. There's also a P site, where the polypeptide is attached to this tRNA. You see that in this cartoon sketch. At the P site, we have a whole polymer of amino acids already linked together through previous rounds of peptide bond formation, and that resides at the P site. And then there's an E site, E for exit, and that's a place for the tRNA to reside right before leaving the ribosome. So that we have, at any one moment during the cycle of peptide bond formation, we have three sites occupied by 3 different tRNAs. An A site with amino acyl tRNA, a P site with the polypeptide attached to the tRNA, and an E site with a tRNA that's not attached to anything. Okay, here is the peptidyl transfer reaction, and it is showing here the P site next to the P site next to the A site. So here's an amino acyl tRNA. We're not seeing the whole tRNA. It's very large structure. We're just focusing on the 3' end. It is acylated with the carboxyl group of an ester linkage between the tRNA and the amino acid. There's a free amino group, and that is going to attack the acyl group of the peptidyl tRNA. And this polypeptide can be quite long. They're showing formal methionine, the very first amino acid, but this could be a very large polypeptide. And then here's the transition state. You'll notice that there's a tetrahedral form, accumulation of negative charge, and they're proposing how water molecules contribute to stabilization of this excess charge, similar to the oxy-anion hole that we saw in the serine protease transition state. But now we're making a peptide bond, and if things go well, the leaving group will be the peptidyl tRNA, so the alcohol group. So this is the bond that's going to be broken right here, and we're forming a new bond between the amino group and this carbonyl carbon, and that bond is shown right here in figure 31.23. In figure 31.24, we see the whole cycle. We see the ribosome. A site is unoccupied right now. The P site is occupied with a polypeptide. The amino acyl tRNA enters. It's being delivered by elongation factor TU. We haven't gotten that far yet in the reading, but that's what carries the amino acyl tRNA to the ribosome. Then chemistry happens, and what we're doing is we're moving the polypeptide onto the amino terminus of the A site aminoacyl tRNA. So it's the polypeptide that's moving onto the amino group, and now we need to ratchet the whole ribosome forward by exactly 3 nucleotides, and that's accomplished by elongation factor G, which hydrolyzes GTP to GDP, so that we get precise translocation. And now we're ready to interpret the next codon. And there's a step here where the tRNA in the exit site leaves the ribosome. Figure 31.25 goes through the elongation factor G cycle a little bit more precisely. You can see here how elongation factor G bound to GTP associates with the ribosome. There's GTP hydrolysis and then movement of this lever arm to reposition the ribosome relative to the A site, freeing up a new A site that can be used in the next round of polypeptide synthesis.